Okay.
Recording has started.
So what we need to, okay, before we continue, I do have some questions here. Okay, Michael has a question. For telephone calls, what is the phone log? What should I do with the email communication on the GLPs? Well, let me see. Can I share my screen? Oh, let me put this one, just block this one first. Okay, you will not be able to see the slides. Let me share my screen again. Okay, now it's myself, share the screen, which is Microsoft Word. Okay. Okay, here's the one. Okay, so let me make this a little bit bigger so everyone can see the words I want to type. Say that short. I do like email. I prefer email rather than a phone log, but a lot of times phone log is necessary, and this also applies to the text messages like SEM, SMS, text messages, or other social medias. This is similar to, let's say, similar to text messages. For example, sometimes it's like Facebook, okay, even WeChat or WhatsApp, okay, etc. So yes, you can do these. Yes, you can, but you need to capture the information. For example, if you called someone or send a text message to someone, you need to write down what you did. Okay, the contents. Let's say the contents of the conversation. So you need to write down. Let me make this bullet. Okay. Then you need to sign and date. So basically, you transcribe all your information from the electronic media, like the text message or any other things, into the paper format. Okay, sign and okay. So in this case, you did that. You sign and date and keep that. This is the work we do. We do have the phone log a form. So I will share this later, but may not be today, but maybe later. I put in my to-do list. So you can use this as an example. You can develop yours, or maybe you can put in your system. So it's easier for you to say check a box, say this is the text message, and this is a phone call. You need to write down just briefly what you need to do, what you did, and what you want to do, and what's the result, something like that. Then you print it out on the paper and sign and date, and that becomes the raw data. So that's the phone log. Okay. So the next question, Michael put it there, is what should I do with the email communication? The email communication is similar. I prefer that. Yes, email is very got a lot of things. You can CC copy other people, and you can get a quick response. And the good thing is you have everything there, and it's built in the system. And all the email conversation communication can be easily print it, and you print it out and sign and date. And good thing is, so far for email communication, that's similar to above, or similar to above. The same thing, you print it out, okay? If you do the email, even you deleted an email, there's no way you say you did not receive that email. Well, you may not read it completely, or you may not understand it, the email, but the server is always there. You cannot delete it from server. Even you delete it from your local machine, from your account, and it's always there. And also the sender will typically save a copy in their the sent box or folder. So it's always there. So in this case, email communication is better because it's easy to track, easy to search, and easy to print, easy to get out. So I do prefer email. So email communication, email is preferred. So I would say that. Okay. So yeah, by any chance, if you can send the email, please send the email. But sometimes you want to do a quick communication, a phone call, a text message, Facebook or FaceTime in iPhone or any kind of social media. That's fine. As long as you capture everything in your work, for example, write it down and print it out, sign and date. So that's how you transcribe your electronic information onto the paper. We will talk about electronic data. I think that will be the end of the last day. I put it there. So how we keep that, we will talk about this a little bit more later. But the overall thing, so far, we define the raw data is on the paper because it's very difficult to validate the electronic data based on our understanding because we need to have the system to validate that. So put it on the paper, sign and date. That's the easiest for everyone in terms of doing your lab work, in terms of doing all those communications. Okay. Okay, let's resume our... Any other questions on this? Let me read the text message there. Who implemented partner country other than the U.S.?
I've already responded to it.
Okay, I don't need to answer that. Yeah, that's... Yeah. Okay, let me share my screen again. Let me put this on the side. That's a big blue screen. I need to stop sharing, then sharing again. And where's the share? Blue. Share my screen. Interesting. I don't see that. It's GLP1 presentation. Okay, I think that's the one. Okay. What I need to do, minimize that. This one we had before. Okay, put it on the side. Yeah, that's good. Okay, this is where we stopped 10 minutes ago. Okay, let's continue. Okay, we had information on this talk. Okay, let me okay. So let's go a little bit further. Well, we talked about the difference, like this definition, sponsor from EPA to, from FDA to EPA. Let's see a little bit more. Let's say this is homework or assignment we do. We try to read the same thing about the OECD. So OECD, we put it there. Well, actually, there's a paper there. There's I can share this paper. It's talking about the difference between OECD, FDA, and EPA GLP. But this is the difference. Okay, the OECD GLP, put it there. is the sponsor means the entity, commission, supports, submits. It uses non-clinical again, the health environment study. So in this case, is the one who is paying that, who is supporting your work, who is supporting the work, and submit. It may or may not be the one who is doing the work, but in the EPA, FDA, it does say, yeah, they can do both. They can do either. Okay, so, but based on the definition, it's probably very similar. There is a little bit difference there, but not that much. Okay, so I'm not going to talk about the differences between the EPA, FDA, and OECD, but I'm just showing there are some differences there, and I will use a couple of more examples to talk about this, and I will come to the conclusion. So let's talk about the testing facility. So based on the words, we know that where you do the study. So the first one, we look here. Okay, this again, this is 21 CFR 58. So we know that this is FDA. This is FDA. Okay, so FDA says that, okay, the testing facility could be person, could be some other thing, and say that clearly is they're doing the work. Again, FDA put it there. It's non-clinical. Okay, so actually doing that. So EPA, now we look the definition. We look here. This is where you can find. It's a 40 CFR 160. So this is EPA. And you will see the testing facility, exactly the same wording, but simply remove the non-clinical. You see, a person who actually conducts a study. Okay, then you explain what is the study, what is testing facility, keep going on. So that is the same. It's only just remove this, the wording non-clinical. So the OECD definition for testing facility, okay, is going a little bit more. Okay, person or premise operates the unit necessary conduct non-clinical again. Okay, then it's getting more like a health and environmental safety study. So based on the wording, you can see that even the wording is almost the same, but using here and there a little bit more. So that is a little bit different. Not that much. Okay. But we will say yes, the OECD, EPA, FDA, if you are using the GLP system, I would say based on what we talked about so far, it's almost the same. It depends on where the focus is. Okay. So we are doing the agriculture work. We are using EPA GLP is good enough to handle all the situations. And this is typically in the US, we follow EPA GLP, but if it is in other countries, mentioning OECD, that's equivalent. Okay, let's do a little bit more before we keep going on some other thing. Okay, let's talk about the QA. So again, this is 20 CFR 160. We know this is FDA. And down here is 40 CFR 160. That's EPA. And again, we read that, okay, here it says QA unit. It's the, all those, okay, as long as this is not a study director, this can be a QA. It can do the work related to quality and non-clinical studies. And the EPA GLP also mentioning that, okay, it's the same except the study director. Okay, so I know you can see the wording is very, very similar. Okay, and how about the OECD? Let's do the OECD one so we can compare. Yeah, what is really the key point? Okay, if we read OECD, it says, okay, that means define the system, the personnel, and it used the word independent. I think the OECD is probably more accurate. I mean, based on my understanding, I think it's more specific. Okay, as long as it's independent, you can do that. Okay, so I think I typically when I do the training in the lab, when I go to your lab to do the work there, I typically will say, okay, is the QA is independent as long as this is not a part of the system actually conducting the test, the work, not the QA is not running any activity in the lab. I did see in some laboratories, some QA, because depends on the workload of QA, sometimes they put the QA to some work. For example, to check the balance or to check the pipettes or something like that. They say, oh, this is not that important. But based on the definition here, if the QA is totally independent, then that is not right. That is not QA. Even though the QA may check other processes, but you have to be independent. So then that is not a QA, that's a QC, because you check something specifically for certain part to make sure the in-house in the system is working, rather than just look outside the whole the QA, the quality assurance in this work. So so far, well, we can keep going on, and we can see more and more. But so far, I think we know now. Yes, there will be a little bit difference between different type of GLPs in the world. Okay, you work in the lab, you do certain part of work. For example, you do the reporting. You may use different terminologies. Okay, so in this case, you just need to stick to one. I will give a little bit more comparison for some wording in the GLP, how this is related to the lab, and how we handle that, how we do that. Okay, so the next one is the SOP, Standard Operating Procedure. Okay, now we see again, this is 40 CFR. We know this is the EPA. Okay, the EPA GLP. Okay, the definition is documented procedure, which describe how performance test activities normally not specified in detail in study plan or test guidelines. Okay, so we can see this. Well, this is the definition. We can read for example, OECD. That's almost the same wording. I don't need to read it again. So, and again, we know this is necessary to follow in the lab. And again, we talk about the study director. So the study director, the 40 CFR, this is EPA, and the 21 CFR 58, this is FDA. So it is the individual responsible for overall study. Just remove this nonclinical, then it's almost the same from EPA to from FDA to EPA. So we know that, yeah, this is just like we give some examples to tell the difference, but there are some other differences there. And also based on our understanding now, it's pretty much we can tell those GLPs are very, very similar. It depends on the details, depends on specific terms. But the definitions may be a little bit different. Let's look at those numbers. I mean, the definitions. For example, in Inception, I already put it there. And in this case, okay, for example, what is the study initiation date? Well, use exactly the same wording. Okay, in all these, that's typically there. Okay, FDA, EPA, okay, OECD, they use almost the same term. Okay, study initiation date, that's typically should be when the study director signs on the protocol. So the next one, experiment starting date. Now it depends, okay? This one is typically defined by the EPA, and it depends on how you do the work. If we were talking about a whole study, okay, so it says experiment start date. So we typically will say this is in the field when the pesticide is sprayed on that crop. So that is the one. But if you are talking about in the lab, it's very, very vague how you do that because you don't know how you do it in the field or something. So we typically don't use the word or use different type of wording, but typically in the laboratory, what we define. We don't know what's going on in the field. We don't know when they spray. Well, of course, you can read the protocol. You can read all the raw data. Sometimes it's not that easy for you to do that. But without this, because there's a GLP clock when you get started, when you need to do the GLP for your study. Okay. So in this case, normally we put it there. Okay. The experimental start date is when you first get the first sample. For example, you have 10 trials from different sites, even from different countries. In the lab, you cannot do anything. So your experiment is not started at all unless you have tested material, you have your sample. So in this case, we typically use in the lab, quote unquote, we use the word, we use to say the first trial received in the lab. Otherwise, you will not be able to do the work. But this is not exactly what the GLP, EPA GLP says there, because that's in the field. So in this case, it's difficult to tell because in IR4 system, the field and the lab are totally separated. So you do the field work, you ship sample to the lab. In some countries, the same people, same set of people, or same group of people, or even the same unit, they do the same thing. They do in the field, they take sample in the lab, they analyze themselves. That's easy for them to handle. But IR4 system, it's a big country. You may get some samples in Florida, like in early spring, maybe in like March, you already have samples. But in the northern Michigan here, or in Wisconsin or Minnesota, you will wait until July to get sample. Even the same trial, same study, same thing. It takes about like 4 or 5 months to get sample doing the same study. So when the lab is receiving the sample, that will be a big span of the time. That could be half a year or even longer. So in this case, it's difficult for lab to do that. But we typically in the lab is the first sample received. We put it there. First, it's difficult to write. Use my mouse. First sample. This is lab. Received. Okay, this is how we define our work, so we know what's going on. Okay. Well, there's another term here. If we look down here, so experimental starting date is a little bit different from here and here. This is the OECD definition. Okay. So, and in this case, it's the date when you have the data collected. So you don't collect data, doesn't really matter. So the data collect could be a lot of things. It could, well, before you spray, you already collect the data. Okay, so even in the field, the dates is a little bit earlier than you actually spray. Because when you do the calibration, when you do planning, you already collect the data. That is the starting point. Okay, of course, the lab part is almost similar because you don't know the field if they are totally separated. So let's read a little bit more before we get back to conclusion. What we need to do, what's the next? Okay, so a little bit more is here. A study completion date. Okay, Now this is the EPA, FDA, GLP. And that means what? You finish the study. It's totally completed. So that means you don't do any work, any activity related to study, except you submit it to the government agencies or JMPR. Okay, so basically is when you finish everything, when the report is signed by study director. So you finish study, you sign this. There's no more data collection. Everything is done. Okay, so you submit it to the government agencies or JMP or somewhere. Okay, the only activity related is submission. It's all done. Then it's completion. Now, here's the thing. The field work, how you say your study is done. If you say you are the PI, part of the big study, and the same thing for the lab, and also for the rest of them, you finish the field work, you finish the lab work, it's not done yet. You are still collecting some QA data, doing some going back review, going back and forth, going communications. The work is not done until study director signs the final report. So typically in the lab, when we think this is the last day, when we think this is the completion day. Okay, of course, this is not a final report. This is your lab report rather than final report. Okay, when lab report is signed, that's typically done. Because when lab report is signed, you will not collecting any data related to the study. However, well, don't take my word that way, but this is exactly the definition by GLP. But in the lab, how we practice is, okay, when the lab work is done, what do you need to do in terms of all your data? You won't collect any new data. The last step is you send the data, you send the lab report to the study director. In our case, is IR4 headquarters. So we send there, we're all done. So when you sign, when the lab director, LRD, we know that, laboratory research director, LRD, signs on the lab report. And once you sign that, immediately you archive everything in your archives. Okay, when you do that, that means it's all done. You put everything in archives on the shelf. I'm not going to do any work. I'm going to submit my report to the study director. So that is in the laboratory. Okay, in lab, when you finish the study is when you do the archives. Okay, so of course the EPA, FDA, GLP definition is the final step. It's like the whole thing. The study director signs on the final report. So there are some other things. Okay, for example, there's still a EPA, GLP. So what is the experimental termination date? Well, this is experimental. This is study. So experimental termination, that means how you collect the data. That's experiment. It's nothing to do with your reporting, your communication, your submission on the analytical summary report or a final report. So this is a totally laboratory thing. Okay, that's why when we finish our study, we only write on this data. We don't write the study. We don't write this. We don't write completion because we don't know when the study director is going to sign on the final report. But we do know when we finished our work. That would be, for example, if you use GC, it depends on your definition. Typically is when the last injection is done in the GC and finish the run, like 15, 20 minutes later, when the data is collected by the GC system. Once it's done, that period of time is your experimental It's done. There's no more. Finished. There's no more. So that is the experimental termination date or something. That's all done, or completion date. Okay. Now there's another term, which is OECD term, which is very similar, but they use the word experiment completion date rather than termination. So in this case, you can see that it's OECD versus the EPA. It's using a little bit word in there, but it really means almost the same thing. So let's say we talk about the GLP, we talk about when we work in the laboratory, okay, when we work, what we really need to do. Okay, we write a different theme, we need to follow different GLP. No, say that short, we just need to follow one. If I train you, I will say just follow EPA. Just follow one GLP, just use one set of definition, one set of the terminology. Don't try to mix them. And that's good enough, because based on what we just talked about, all other countries or other GLPs will accept, and EPA GLP is accepted almost everywhere over the world. So my suggestion is use one set of GLP, EPA GLP, for example, and stick to it. Don't make the change. Okay. So, well, the GLP, we just go back to the EPA GLP. It says it's the regulation. That means it's the law. Okay, that's the law to ensure the quality and integrity of the study. Well, we put a non-clinical there. That's more broad definition for both EPA and FDA. Okay, then we talk about the what is the law? Okay, law is the law. Well, you know what that means. Is that right? So if you don't follow the law, we know that means if you're breaking the law, that will be sent to the jail. So that's clear enough. I don't need to explain the detail or more about this. And this picture is used almost every single time when we talk about enforcement. Okay, so you work in the lab, you don't follow GLP, but you sign the GLP compliance statement. That's a big thing. So you follow GLP, you put it there. You don't follow GLP, you write the exception in your work, and what is there. Okay, for example, this step is not GLP. Okay, and that's quite typical in certain times. For example, we use just an example. Your internal standard is not GLP. Okay, you just get it somewhere, and it's no GLP certification showing your internal standard is GLP. And in this case, when you finish your report, when you sign that statement, the compliance statement, just put that word there. Okay, everything else is GLP except the internal standard is not GLP. You have which source get it. That's it. That's good enough. Okay. So that's easy enough to make it straightforward. Okay. So how about we do different work? Some labs, well, IR4 lab is very straightforward, very simple, because we only do the GLP work. We don't there's no other work. We only do the pesticide residue, the GLP work for IR4 studies. That's in our lab, so we don't have this problem. But if your lab is doing different work, then we need to figure that out. Which part is GLP, which part is not. Okay, so we just listed there the basic information about the GLP. So anything related to pesticides, registration, health, and the environment, EPA requires your work should be the GLP. So all the activities, for example, if you are planning, okay, you write something, oh, I want to do this, that must be GLP. And you do the work, you do that real LC-MS analysis difenoconazole. So from the starting point to finish the study, you need to follow the GLP. Okay, and there's a GLP monitoring. So in this case, should be both QC and the QA. QA is totally independent. So that part should be GLP. And all the data recording, all those things, you should put in a GLP format. And EPA may come here to audit your work. And the reporting is also GLP. So you need to go through inspections, go through the QA or extend inspections. Then will be, for example, study director need to get all those data organized and archived before you submit, before you sign the final report and submission. Okay, so some other things here. You do not have to do GLP if it is not related to any of these. For example, if you are doing non-regulated, you are not going to do any related to the regulatory. For example, you only do the scientific finding. You work in a research lab. Okay, right now, coronavirus, okay, is very hot topic, and a lot of people doing that work in the research, scientific research, not regulatory work. Then you do not have to do the GLP, and the GLP does not regulate any of the science. So GLP does not regulate science. Science is science. And even you do the GLP does not mean you are doing good science. You can do a very good GLP, a lot of documentation there, does not mean your science is good. So the GLP does not regulate science, anything related to scientific research, as long as this is not for the regulatory purposes, you do not have to do GLP. We can do some other thing. Okay, now we're talking about different things. So we talk about the agencies or different organizations. We did talk about the different GLPs, for example, FDA and EPA. Okay, and they will review your data. Okay, and this is also result in some other work like safety work or efficacy. So it depends on your work. If you are working mostly in the laboratory, we can just go on the next. Okay, do we have any questions? What would be the audit in India for GLP?
I've already answered that.
Oh, okay. Thanks, Michael. Yeah. Okay. Let's just move on. The next. Okay. So this is just to go back to what we just talked about. Okay. We talk about, well, make it a more broad way. We talk about the clinical, non-clinical, make it easier. Basically, if we are talking about the pesticides, agricultural work, you can simply remove all those non-clinical there. That's typical, but in some studies could be mixed or could be there. So just leave it there for general purposes. So the GLP will do all those things. So if you do the safety, you need to do the GLP. Okay, the pesticide safety. Okay, and you need to do the agricultural pesticide development. That is GLP. Toxicology work. That is GLP. Food and food additives. That is GLP. And test the all those hazardous material. That is GLP. So this is what we say. It's more broad way, talk about areas. Okay, so if you are working in the lab, it depends on your system, But if we talk about the regulatory, talk about the EPA, talk about the JMPR, the pesticides registration, yes, they're all GLP. Okay, this not necessary for GLP. We mentioned anything is basic science. Okay, if you do basic science work, you do not have to do GLP. And in this case, there's one thing. This is exempt. Not necessarily you do not do GLP, which is because when we do the GLP work, we do the method development. There's a lot of things we have to try this, try that. That makes things very complicated. For example, your method development, you work in the lab, you work on difenconazole, mango, okay? And you spend a lot of time working on that because there's a lot of sugar and the fat and a lot of wax. The reference method just does not work. So you are working on that. You spend a month trying to do method development. You are developing a method, and it's not finished. It's not a finalized method. So in this case, we call that it's exempt from GLP. It does not require to follow everything GLP, but you are expected to keep everything. Because when you go from step A to step B, why? Then you need to have the data to show that. You do not have to include in your report to do GLP. For example, if this is a study, you do the mango study, difenconazole. It's just, you keep trying that, it doesn't work. But you use different kinds of salt, not necessarily the same thing as in the reference method, it worked. So you can say, yes, I use that, but that's a change. A lot of times, study director may say, no, this is not good. You cannot do that. But sometimes, yes. And sometimes use a different method and make it work. It depends on the situation. We will go through this later in the next several days. If we are talking about the GLP and the science work, if it doesn't work, it doesn't work. Okay? If it works, does not mean the study director will accept. So that's why when you do your work, you need to talk. Okay, you need to talk to the study director. Is it good or not good? So in this case, it's only the method development is called exempt. Okay, it's exempted here. Okay, it's not necessarily non-GLP, but exempt. Normally, EPA does not require to read the data, but they do come here once in a while to check. It's not just say from your head, just find it somewhere, or you cut corners. No, you have to show people you did this because it doesn't work. Oh, I do this, it worked. Okay, so that's one thing. If you are not doing any registration purpose, for example, if you are talking about other thing, they are derived from like a market related, that's fine, as long as it's not registration related. Okay, it's not that part, you do not have to do GLP. So three things, are we clear enough? If it is basic science, that's not a GLP necessary. Even you do the GLP work, in some portion, typically it's very initial part, you have to do GLP, but this is typically called exempt from that part. So in my training, I typically say, okay, this part, method development, okay, before you really get to the method validation. That's fine. Okay, so you can leave it on the side, keep it in a separate pile of your data. Or this is market-related, non-registration purposes. That's fine. Okay, so that is that part. So the final conclusion. Okay, when we talk about those GLPs, we talk about typically three types of GLPs: FDA, EPA, and OECD. Okay, are these compatible, or if you do EPA GLP, will this be accepted in Japan, Australia, or EU, for example? So my experience, my comment, I can tell, yes, they are compatible. They are equivalent unless they have very specific requirement for that purpose. Okay, for some of them, in their protocol, they may add special thing. For example, I talk about protocol. For the lab part, okay, and EPA does not require to do multiple injection or multiple type of standards when you do the calibration. But some other countries, they put it there in the protocol. Sometimes that it is required in their work, and they call that a GLP. For example, if we work with the NAFTA, with Canada, we have to add our own more requirement because Canada has different requirements. So this is something depends. Now let's go back a little bit. So it depends where your data are used. You submit it to Canada, you have to follow their rules. You have to follow their GLP plus all the government-related regulations. Okay, but if you submit it to your own country, you follow your own country requirements. And they may or may not be overlapping in certain way. Requirement be these are all equivalent, but they have some other special thing. So read it carefully. For example, if we do the work, we have about 10 or 12 studies, combined studies with Canada. So because Canada, it's just like extended U.S. zones. We got 13 zones in the U.S. And in certain way, for example, in Michigan, we are zone 5. And in Ontario, it's also zone 5 because we are very close. In this case, I should show you the maps. Then it's easier for you. Let me just show that. Let's make it easier. Okay. Can I share the screen? Okay, maybe I can. Okay. Let me try to share the screen, see how that works. Okay. Oh, it does not show. It's back there. Okay. Let me see. Stop share. Then I share again. Share screen. Then it's Zoom. Okay, it's here. Do we see that? Okay, yeah, it's here. Okay, this is Google Maps. I'm here in Michigan. Michael's here in New Jersey. And most of you are in this area. Okay, so that's Asia. So it's 12, 13 hours difference between Michigan, New York, New Jersey to Bangkok, Singapore, Malaysia, all those countries, Pakistan, Nepal, all those countries. Sorry, I can't name all of them. Okay, let's go here. We will see. This is Michigan. This is where I am located, right? Michigan, you see, it's just like this. Okay, this is all like that. Okay, and this is zone 5. Zone 5 is this region, and Ontario here is also zone 5. So in this case, our trials can be shared because it's the same climate, the same geographic zone. And you will see this is Great Lakes. Okay, do you guys know the names of those lakes? Just say something a little bit. And Lake Superior, I think it's the second biggest lake in the world. Lake Michigan is Lake Michigan is I think number 5 or number 6 biggest one. This is Lake Michigan. Okay. So Superior, Michigan, Huron, Erie, and Ontario. And Michigan, this is a Michigan, Lower Michigan. This is Upper Michigan. So Michigan is surrounded by 4 Great Lakes.
Recording is on.
Yeah, except the Lake Ontario is away from Michigan. And Ontario is the same, but except the Michigan. So it's Michigan, Ontario, Ontario. So Ontario is surrounded by Superior, Huron, not Erie, not Michigan. Actually, Erie is here, Erie and Ontario. So it's easy to remember. Well, in elementary school here, to teach kids to remember the Great Lakes, the name. It's very simple. Now you can see this is Superior. It's super. Michigan, man. Huron, H, help. Erie, every, E, Ontario, O. So 5 Great Lakes is Superior. That's Superman, super, man, helps, everyone. So that's a little bit thing for you to remember those 5 Great Lakes. Okay, Superman helps everyone. 5 Great Lakes, and this is Michigan. This is where I'm located now, right in the middle of Michigan. So this is the time zone. The Eastern time zone. This geographically, this is at 5, and in this case, we share the trials in many studies with Ontario, sometimes Quebec. Okay, so if we do this, we need to follow both the U.S. EPA GLP and requirement and the Canadian requirement. Do you know where the Niagara Falls is? We are very close to Niagara Falls. Yeah. So we are here. We drive about 5 hours here. Niagara Falls is here. Right here, Niagara Falls. This is a Horseshoe Falls. Okay, Niagara Falls here. And okay, let me just go a little bit. Okay, this is where I studied and lived almost like 7, 8 years. It's about half an hour to Niagara Falls. Toronto is here. And this is where I showed you a few minutes ago where I worked. It's downtown Toronto, which is right here. Yeah, downtown Toronto, right here. So basically in the past almost 30 years, 25, 26 years. No, longer than that. 29 years now, 28, 29 years now. I lived in the Great Lakes area, just from Toronto to Michigan, just all around the Great Lakes. Okay, that's something more than that. Okay, let's go back to our slides and share my screen and slides here. Okay, we are back here now. Okay, so we say international GLP. We know that they all accept each other. Okay. So in this case, we don't have to worry about our work will not be accepted by other countries. So I would recommend, because I'm doing the laboratory GLP training with the GLP with your lab practice, I would say follow the EPA GLP. That's easier for me because I use the same term. I worked in this system in the past 20 years, so that's easier. But you work with under other GLP like OECD or other countries specific GLP, that's fine. They will accept each other. Okay. So yeah. Okay, let's go back to the keywords we learned in this. We learned the keyword testing facility. Okay, we learned study director, of course, that's the single point of control, which is very, very important. The these are the federal food law in the U.S. and FIFRA. And okay, I leave this NAFTA here because it's North America thing. This is only U.S., Canada, and Mexico. We learned the word sponsor, which is the boss, okay, who pays you to do the work. That's easy to understand. And what is the raw data? Typically, it's paperwork. And if you have any like electronic data, it's better to convert it into the paper, sign and date. That's the raw data. For example, the text message, the phone call, even the email, you need to print it out, sign and date. That becomes the raw data. And there is something we will talk about like a PI, which is right after the study director, which where's the study director? I should have put SD, we had the previous one. So this is under. So for example, if Michael is the study director for a study in Thailand, for example, and because a lot of times he works in different projects, he also need to work in the U.S., and a lot of work need to be done in local countries, for example, in Thailand. Then he can assign a PI there, okay, principal investigator, so can do a lot of work there. We know in the GLP we cannot have two study directors. No, we cannot. We can have another people to help to do the work. We typically call that PI. So we learned today QA, QAU. We compare the definitions in FDA, EPA, and OECD GLPs. We also did some SOPs. I mean, overall thing. And one of the very important job for QA need to do is to keep the master schedule. That means you know how this is working and what did in the past and what's doing now and what's planned for the next. So study plan, protocol, that's the same thing. Then we probably will mention something, amendment, deviation sort of things later. But I think, let me see, I have any questions? No. Okay. Required for study. Okay. Yeah, I think Michael answered most of the questions there. The questions in the chat. Thanks, Michael. So, any questions? Anything? So before we go next step, this is typically we do. Okay, I do when I do the training. Okay, when I do like a half an hour or so, then I stop at like 3 or 4 minutes, ask questions. And if you don't ask questions, I will ask you the question. So it's better to have the question to discuss first before I ask you. Otherwise, you'll be, yeah, you know, yeah, those people who went through my training know that. Yeah, that's sometimes it's getting longer, but anyways. We still have a little bit time, so we want to talk about. Let me pull out this one. Any questions in the background? Accreditation, Michael already answered. No, looks like that's okay.
Yeah, Wayne, if I could just make a point.
Yes.
Yeah, so there are kind of different levels within GLP. So there's the way in which GLPs are practiced, and then there's also the infrastructure of GLPs. So the infrastructure for GLPs also goes up to a country level. The reason I mentioned this is that Wayne has correctly stated the importance of adherence to GLPs, and we are explaining how to conduct studies under GLPs, but obviously each country we are cooperating with is an individual sovereign country, and most of you do not have a GLP infrastructure yet. Okay, so when Wayne was reviewing the section that GLPs do have a legal implication, that is true. So, for example, under U.S. EPA infrastructure, we have to sign that we are claiming GLPs, and that there are legal implications. Yes, you can potentially go to jail if you are flagrantly violating GLPs. But I just wanted to make sure everybody understood that we are training how to conduct studies under GLPs, and each one of you have a sovereign country that you're a citizen from, and you each have your own laws. So don't be concerned that there's some legal implication unless that's within your own country. So whatever you follow from that aspect is within your own country. But in reality, we are training you how to conduct studies under GLP. The whole infrastructure really has to be developed within your country, and that is a process within itself, and more likely under OECD.
Yeah, thanks, Michael. Yeah, I want to add something to this. Yeah, well, let me tell you a story. When I was doing a training, this kind of lab GLP training in a country, I don't name this country. Yeah, it's not in Asia. Interesting question is similar to this. I put this slide on, everyone just opened eyes like that. Oh, yeah. But suddenly there was a joke. Somebody said, oh, send people to jail? Are you going to jail me in the U.S.? And everyone laughed. They said, oh, that's good. I can go to the U.S. That's good. Well, I said, yeah, well, that's not a necessarily true, but in the U.S., this is the law you have to follow. And if your product and if your data are used in the U.S., you have to follow that. Even not in the U.S., it's related to U.S. You have to do that. You have to follow that. Otherwise, there will be some kind of a punishment, but not necessarily like what Michael said, to enforce in your own country. But it's in a different way. But I don't guarantee they will jail you. They will bring you to the U.S. and send you to jail. But I'm just saying we do our work, and this is the requirement. Yeah, that's a joke. Yeah, actually, that's the true story. They did ask the question. Then some people say, "Oh yeah, you bring me to the U.S. That's good, so I can stay in the U.S." And well, that's a different story. Yeah, I agree. Yeah, Michael, thanks.
Yeah. Also, I want to add that the thing that usually triggers this is enforcement actions are involving data that is submitted to an agency, like submitted to EPA or to FDA, etc. In this particular project, this residue mitigation project, the data is not going to be submitted to any regulatory agency other than if you choose to somehow use it internally. Thanks.
Thanks, Michael. Okay, let me see if I can. Oops. Why it's showing on this screen? Let me change that. You are sharing. I was trying to share, pause share, stop share, pause share, stop share. Then, okay, I got a wrong one. It's showing my presenter screen. I need to show the other one. Let me show square. Okay, show this one. That's still the wrong one. I'm sorry. This one, this one. That's a big blue screen. Okay, okay. That's it. That's the one I want to see. Okay. Okay. Why it's stopping there? It's moving back and forth. This is a little bit lag on my computer. Well, I used this slide last week in the inception, and I want to just go through this a little bit. I want, well, I know a lot of audience already sent the information about the availability of the equipment you have in your lab. And in this case, let's look this one first before.
Wayne, right now yeah. I see a blue screen.
I see that.
Oh, it just flashed.
I know. Why it's not going there? Let me see. If I go.
Okay. Now I see it. Laboratory equipment. Oh, now it's gone.
Why this one is not showing? Interesting. It's the same slide.
I see the slide now.
I know. Let me
it's
just
like
go up one more. Up one. Table. Yeah, this is what I want to say. Okay. Well, let's see. Let me use this pen. Okay. Let's look the column. This column here. Okay. We have all the chemicals here in this column. That's the list, and we may add something to it. That's fine. Let's look currently. Look those chemicals plus those chemicals. And we pretty much sure, based on our knowledge, based on our experience, and we pretty much sure LC-MS should work for all the chemical or the pesticides on this list. So if your country, your lab has the access, has the this equipment, we pretty much can go ahead. We have the green light to go to do the work. Thanks to Dr. Wu, did a lot of work, and I also did a little bit on this. Some of the equipment, for example, HPLC or GC, most of them, especially for GC, like FIDA, MPD, may or may not work, most likely will not work very well with low level. So if we have the LC-MS here, we pretty much know we have the green light to do the laboratory work. And if we have some other things like in the middle, like HPLC or GCECD or something, we may be able to do a bigger list of this, but not necessarily to do all. So it depends on the situation, depends on the residues. If the residue is very low, typically because we do the mitigation, we expect it to go down to very likely, Michael, if I'm wrong, please correct me, it's 0.01 as the detection limit. That's ppm.
That would be the goal, yes.
Yeah. So unless we do know the residue. So if it is like that, the GC will have very much difficulties to do the work. So yes, we can do cleanup, we can do the concentration, For example, GCECD is very sensitive, but it's also got a lot of interferences there. So it may or may not work. So that's a big question mark there. So I know some of you already sent information out. Please send me the email if your lab has the LCMS or some equipment we do the work. For example, even GCMS, most of the list should be okay, except some of them because they will not be able to be vaporized. So that's something we need to do a little bit work, and also in the same time, we can think about if you do not have the equipment, is it necessary your lab can do the work or can be that low? Do we need to develop a better method, spend more manpower to do the work? For example, to do more cleanup, and if we do know the residue, for example, if we know the residue is not in this range, if we know the residue is nearly 0.1, this is the real residue number, then we make our life a little bit easier because we do know what's going on. It's difficult to tell this time, but it will make Michael's life, the study director's life, much difficult because once you see the higher residue or higher detection limit, something will be difficult for them to handle. But anyways, we do the best we can. So I do need some information, and I probably, hopefully, when everything is going okay, I'm thinking to visit your labs. I visited several labs there, Thailand. No, I did not visit the lab in Vietnam for this purpose. Visited the Philippines. It's not on the list, but yeah, the rest of them I did not visit. Well, I don't need to visit Malaysia. Malaysia is quite good, and Singapore is quite good. I worked with them close enough in the past. So yeah, I probably will visit some of the on the list on the side and see what we can do, what we need to do. Okay, so I did a little bit of homework. Which country has the equipment? Which country may or may not? So please send me email. I can finish this list, and I will plan my visit accordingly. And also, I did talk to Michael. Michael sent me some email, very good information. It depends on the method. And most likely we're going to use the catcher's method. So in some countries, first of all, the availability. I know when I was in Africa, they order some material, and typically those materials are from the U.S. It takes about 3 months to get those materials. So that's really long time. And not only that, the price is also 3 times more expensive. So, well, if I visit your lab and we take the advantages, I can bring some materials there. For example, those are catcher's material. Okay, so we can do some work immediately, and also will be cheaper catchers. It's just like a first, second grader writing something. Okay, so email me that what your the method will be, and what your work is, and what your instrument access. So we plan this, and also if you do need some help on those small items like catches, or even some small hardwares like something I can bring there, I will help. That makes things easier for you, and it's easy for us. We order something, we just make a phone call or do that online. We can get the material in my lab second day. Not paying a huge the shipping. The shipping is probably $5 or $10 extra. So that makes things easier for everyone. So send me email and any question I can answer. Surely I will do that. Okay. Any question about the equipment, about method, about those things? We still have about maybe 20 minutes to answer the questions. So please send your questions in the chat box so I can read that. Michael can read that and can answer the questions. Okay. EOA Thailand. Oh, I know that you have both GC, LC. Okay, that's fine. Please write in the chat box so we can answer the questions.
Yeah, so especially for the developing countries that were not part of the previous STDF project, like Bangladesh, Nepal, Sri Lanka, Laos, Cambodia. If you can forward your analytical equipment specifics to Wayne, that would help a lot in understanding our approaches.
So you know my email, eh? It's very simple to remember. My last name plus 2 letters of my first name at msu.edu. So you will see my email on every slide I have. And I will, it's a mail list there. I can email those slides I talked today. So I intentionally keep the slide here without sending to you. Otherwise, you guys will sit in there doing nothing, just watching that. So if you don't have anything to watch, you have to watch the screen as I am talking about. But I will send the PowerPoint slide to you after this. Okay. Questions on there? Bangladesh, FGC, FID, ECD. FID, very likely, as we showed on the previous slide, may not be good for this purpose because our detection limit is relatively low. So we will see that FID on this list may not work very well, almost for most of them. Getting to this detection limit will be very difficult. ECD is a big question mark. It depends on the matrix, depends on how much halides in the compound. For some compounds should be fine. If you do a little bit cleanup, make sure there's no interference. It's ECD. I used to remember I showed you my Toronto work experience. I was making a detector, and that detector is ECD. So ECD can be very, very sensitive, and if you can make sample clean, ECD can be equivalent to GCMS if you only look those halogens like chlorides, fluorides. It's equivalent, but with interferences, with those things, ECD sometimes is not good at all. And the ECD, the bad thing is it's because it's a radioactive. Okay, it's going down every year. So maybe last year it's very sensitive. This year it's getting worse and worse. It won't go bad like in the next 5-10 years, but it's slightly going down the response. So that's fine. But GCMS, well, it's more expensive. So you have to try if you have the ECD or GCMS, see how this list works in your lab. I don't know, but we have to try. We don't know yet. Okay. Well, derivatization, I cannot tell this time. It depends. Sometimes yes, but derivatization, the biggest thing is the recovery. Sometimes even it works and it's not that complicated. Takes probably like another couple of hours, but the recovery is bumping up and down. So unless we have to use the GC method, it's better to use the LC to handle that. That makes much easier, and you get more consistent results. Okay. Okay. Let me see if I need to go any further on this slide. I probably don't need to, but okay, this is just where we stopped. I can make this a little bit smaller. We did all these. That's fine. We did all these. We don't need that. Yeah, I don't want to go any further. To go next, to go through the inspection details, because I do want to go over that a lot. This is a lot of people asking me the question, how do I do this? How do I need to prepare for the inspection, for QA inspection, for EPA inspection? Okay, which LC-MS you have? Michael, is the question you ask me or you ask Sri? Okay, that's sorry. I think they have the waters there. I went to their lab. That's waters. Okay, is it possible to pesticide residue mix?
Recording is on.
Okay. Well, the GLP does not require you have to do a proficiency test or something. ISO 17025 requires that, but GLP needs the full validation. So my answer is no, you do not have to do the proficiency test for the GLP work. You have to do the full validation. But if you do the ISO work, maybe a different thing, because they need to check your specific of the method, matrix, and the work. So it's different requirement. No. Okay. Another one. Well, I'm reading my laptop here. That's why you don't see my face there. Okay. Okay. In Cambodia, we have the FPD, MPD, and HPLC. Well, HPLC is difficult to tell. It's not on the top of the list. If I choose, I would choose number one is LC-MS for sure. That's number one. That's no doubt. Everyone knows that. I prefer that. GC, MS, even the GCMS will be number two. I choose. HPLC will be number three. Then the rest of GCs will be number four. If I have to choose the order, I would choose this. So if it is in this case, I would say try on the GCMS if you have. We can work on the LC. The HPLC, it's difficult to tell, and some of them may not be very well, unless you have very good absorbance on that material. Yeah. So even for some very difficult matrices, you can, the compound even like a paraquat, diquat, you can use HPLC, but it will make your life miserable. It takes about 3 days to do the cleanup. So yes, it's possible, but it's better to use a different one. So it depends on your instrumentation. So send me an email, please. Okay, other questions? Okay, Nepal, we have GCMS. I think GCMS, you look on the list here, GCMS can do a big list there. Maybe the only thing we have to think about is can this chemical be vaporized? Is it compatible? Once it's vaporized, I think we get 90% chance the GCMS will work. Yeah.
Yes.
Okay.
So Wayne, if you look at the table you have on your slide there, so for Cambodia, they're doing chlorpyrifos and cypermethrin on basil, and then tricyclazole. So you have in this table you have yes listed for either GCMS or GCECD.
Yes. It's here. Yeah. Yeah, that should work. Yes, that should work. Yeah. Well, even ECD with chlorpyrifos should work. Cypermethrin may or may not. This is a little bit complicated if you use that even with HPLC. Typically we use the fluorescence, but I would say GC will work. GCMS will work. Yes, that will work.
So there's another question from Bangladesh saying, is LCMSMS mandatory or not?
My answer is no, it's not
Bangladesh.
mandatory.
Yeah, so if you look under the list for Bangladesh, the active ingredients they would be interested in are in greens are acetamiprid, imidacloprid, and malathion.
Okay. No, LCMSMS is not mandatory.
So, but according to your according to the table here for imidacloprid, there it says GCMS will not do imidacloprid.
Yeah.
And neither will GCECD.
Probably I have not checked. I should have checked HPLC. Maybe HPLC will work. I don't know. We have to see.
Okay.
Try HPLC. Yeah. Well, they can do a little bit of homework, and I can do a little bit as well. Try this one. It will not work on the GCs. No. So yeah, you can do derivatization, but that's making open the door to another set of questions. The efficiency, the recovery. So that makes a little bit easy. Yeah, I mean, difficult for us to say again because you get to the next step. It's difficult to tell, but it's workable if you need to do. Yes, that's fine. There's other questions. Okay, just comment. Pakistan not finalized at the screen. Okay. Yeah, Michael can answer that question.
Your statement is correct, and I understand you're leaning more towards rice. So this table was developed a while ago. So based on the information we had, we thought maybe you'd be working with greens because you have BT, but maybe we will be looking at a different crop. And so you're correct. Yeah, that's my understanding as well. The crop has not yet been finalized in Pakistan, although I understand in a general sense your top priority might be rice if you determine that.
Mm-hmm. Well, the list may not be 100% correct on this list here.
What
the
active
instrumentation.
Ingredients. Yeah, because some of these are changing a little bit.
Yeah, it's a moving part, but that's the overall thing. Overall thing we have, and the same thing for the methodology for the instrumentation. This is based on the experience. This will work, and it depends on the matrices. Sometimes you may spend a lot of time to do the work to make it work. It's hard to tell, but that's our experience. And yeah, both I and Dr. Wu spend a little bit time on how this will work, which one will work. It's not a complete list. It's not necessary. Say the equipment is must for for this work, but at least that's the common sense. If we are in this area, we will make things easier to do the work. So yeah, depends on the lab situation, depends on the chemicals, the crops working on. We will have to work on them accordingly. And if we, for example, if we do have to do, you only have the GC in this case. We have to do derivatization. We will do it. Okay, it's just like a preference. I've been working with LCMS, MS in the past 20 years. I really recommend LCMS. That makes your life easier, make my life easier, because when I do the training, I can directly go there, do this. Maybe I just need to spend 3 days with your lab, and pretty much you get the sense where and how you do the method validation, and hopefully to help you to do method validation. So that's the goal we have. And if you have to spend more time on the analysis, process the samples, clean up the samples, then that will be a little bit more work. I may spend, if I visit your lab, I may spend another couple of days more, make sure you can do it correctly. I mean, in a certain way we expect you should follow. Okay, other questions? I want to take advantage of your expertise. Ask this GC, amenable pesticides. Well, this is the same thing we keep talking about. Is it a GCMS or GCECD or some other ones? Yes, GCECD depends. Okay, this one, that's why I say I used to work in that company. I'm making this ECD detector in Toronto. It is very sensitive, ECD, but you have to make sure the background is cleaner as well. Not like the GCMS, maybe a little bit better. You can have a little bit dirty samples. You still can get very good sensitivity. But with ECD, sometimes we use the term called that a memory effect. If you inject a higher concentration of bromide or chloride compound in the system, and that chloride or bromide will stay in your GC system like a ghost peak, and you don't know when and how. So if you can handle that, you can clean up sample very well. ECD will work equivalently to GCMS. Okay, if you have the chloride compound or bromide compound. So it's tricky, it's sensitive, but you have to work in the right way to remove the interference. Okay, the interference is there, then you see the ghost peak, you see the background, then it's not good. So it is good to do, yes. Okay, what is the extraction method for pesticide catchers? I would say at this time, I'd recommend the catchers unless you submit to EPA, because the EPA is very restrict on the initial step of the extraction. So if our work, okay, for example, if our work here submit to EPA, I will say no, depends, don't do it. Okay, I will say that. But for our JMPR submission, Michael, will tell you the true story about the next step. I would say yes. My answer is yes. So yes, we use catchers. Unless it doesn't work, we have to change a different one. And I know catchers is a very standardized method. You have the catchers salts and acetonitrile. You do the extraction, and cleanup may take a little bit time, or a different thing, depends on how much protein there, how much sugar there, how much fat there. So sometimes you have to do different cleanup. If the matrix is just like some letters or something, it pretty much, there's no other cleanup need to do. Just very simple, maybe DSPE, shake, centrifuge, and you can inject it directly. But with like a basil is difficult to do. Pepper is difficult to do. There are a lot of things there. So you may have to think about more about the cleanup, how you do it. But we will work on that. You need to do a little bit method development, and well, at this time we do remotely. And I tell my experience, we can have a conference calls, Zoom meetings, Microsoft Team meetings, so we can talk about that. And when the travel ban is lifted, I'd like to see physically in your lab how you do the work, how the instrument is working. So I do have quite a lot of experience running those things, but depends on sample different from matrices. I don't promise and will make it work immediately, but I do know the trend how this works. I worked in the system long enough in terms of GLP, in terms of chemistry, and I experienced it, different like up and downs doing in different labs. So my answer to your question is yes, we do the catches, but it depends how this works. Okay, I think it's almost the time to get to those things. I will save that as my record. So I mean, just simply, how can I do that? Yeah, how can I save all those chat then? Click to copy. Anyways, I will do that. And send me the emails. Okay, I will answer email. I know maybe this week I may not be able to answer every single email. I have the whole week, and also, yeah, I have other things to do, but I promise I will get back to you. Okay. Yeah, it's very, very good. Those are very good questions. Actually, when we really talk about the lab, talk about extraction analysis, you will have more questions. This is my experience. Teaching like this kind of a training or teaching, we don't learn things that much. Okay, I went through a lot of trainings myself. I trained myself. Yes, it helps, but not that much. And really the communication within the training, after the training really helps, because I have a question. In the training, the trainer, let's say, for example, I ask EPA, like Francisca Ling, she may give you a whole bunch of GLP. You don't learn things because, yeah, it's so complicated. But when somebody asks a question in your lab, how you do this in terms of the GLP requirement, then the answer is there. Then the trainer gives you the final better decision. That's much easier for you to handle the work. So I learned GLP this way, and same thing. When I train you guys, I learn myself. I'm happy about that. Okay. I know it's some places like it's quite late. It's like almost 9 p.m. there. Sorry about that. Keep you so late. And right now it's so, yeah, I can see it's very bright outside. Yeah. Okay, send me the emails. I think, yeah, see you tomorrow, same time. We will go after that. You have the, did I send you the?
Wayne, there was an
agenda?
additional question.
Yeah. Oh, this one. Okay. Only GCMS need to learn derivatization. What do you think? That's a good question. Depends on the chemistry. We may not be able to handle in the training here, but after this training, we communicate and see what we do. Most of the derivatization is methylation. That's typically do that. You add it to it but we'll talk about that later because it's complicated and it's getting more detailed chemistry. I'd like to share my screen and do something. Yes, I do have quite a lot of experience on doing that, especially for those water soluble herbicides. It's very very tricky to do. Without the derivatization, your recovery is very poor, and it's very matrix dependent. Sometimes if it is matrix is clean, no cleanup is needed. Even you don't need, even it doesn't work with GC very well, it still will get a good recovery. But when you change the matrix with the fat, with the lipids, it's totally upside down. So yes, I will. Just send me an email. We will talk about this later because it's getting too much for the short period of time to the training. I'm sorry about that, but yes, we will work on that. And yeah, hopefully, if I go to your lab, I can show you some more steps and experience that I have. Yeah, thank you. Okay, Michael, you think it's good enough? It's almost 9 o'clock, and should we stop here and continue tomorrow?
Yeah, that's fine. So thank you everybody for your attention and for some great questions, your participation in this first of the series of the lab trainings. So there will be the next lab training will be tomorrow, the same time, whatever your local time is that you started today. It's the same time tomorrow. The link is going to be the same. So no need to worry about the link. Again, just continue into the blue jeans as you did today, and it should be ready to go. And Wayne, do you have any other comments?
No, that's good. Because I don't want to continue. If I continue, I will take too long, and I think that's good. We will go ahead tomorrow to continue doing the training, doing all those questions. Yes, and I want to do that's related to like a QA forms sort of things like that. That because a lot of people ask the question. Yeah. Thanks, Michael. No more questions.
All right. Well, with that, I think we are then concluding today's session. We look forward to your participation tomorrow. And for most of you, have a good evening.
Yeah.
Bye-bye.
Okay. Good evening. Bye-bye.
Good evening.